Hello and welcome back. Today is all about membrane transport. So we're going to talk about the different ways that cells move ions, nutrients, water in and out through the cell membrane. So let's get started with just some really basic key terms that you're going to want to know because these are going to be in definitions moving forward, and if you don't know these words, then you're not going to know those definitions. So when we're talking about a solution, solutions are made up of two parts. They're made up of solutes and they're made up of solvents. The solutes are dissolved in a solvent. So if we were to make Kool-Aid, the solute would be the powder that comes in the package and the solvent would be the water that you mix it in. If you were going to add sugar to water, the sugar would be the solute, the water, again, the solvent. Then there are two main types of transport. There's passive and active, and the main difference is whether energy is there. So passive transport is no energy, and active transport needs energy to be able to do the transportation. And then when we say high to low, like something is moving from high to low, it's moving down a concentration gradient. So in this picture over here, right, 50% salt solution to 20% salt solution. 50% is higher, 20% is lower. So it's going to move from the higher amount to the lower amount. So we are moving these salt particles across the membrane, right? That's this like dotted line, but it's moving from the high concentration to the low concentration. So it's moving down the concentration gradient. So, main types of passive transport, there are three types. These are two of them. No ATP. Remember, that's the big thing about passive transport is they do not need energy to actually transport molecules. So, diffusion is the big form of passive transport. It's the movement of molecules. Can be anything, salt, sugar, ions, anything. Movement of molecules from high to low, just like we showed on that previous slide, movement from high to low, and then a higher concentration gradient leads to an increased rate of diffusion because if you have a higher gradient, meaning that there's a bigger difference between the two sides, then the molecules have more energy and are going to move more quickly, and because they can pass through the membrane without assistance, they're going to just move until they hit equilibrium. Facilitated diffusion is the exact same thing as regular diffusion, but instead of just moving freely through the membrane, these particles are a little bit too large, and so they need to go through a carrier protein to actually move through the cell membrane. And remember from the last set of notes that the cell membrane is made up of a number of proteins, but one of them is a carrier protein, and it is used to allow transport of maybe like bigger sugars like disaccharides, starches, stuff like that. So they're very specific. These proteins are specific to the molecules that pass through them. But again, no energy is needed, so they can pass through pretty freely, but they just have to go through the protein rather than just slipping through the phospholipid bilayer. And then the last type of passive transport that we're going to talk about is osmosis. This is a very particular type of transport. This is strictly the transport of water. Again, we're moving from high to low, but we're only moving water. Only water. That's all osmosis does, is move water so that the concentrations become equal. So in this image here, you'll notice that this membrane here is really small. Like the openings are really small, and these solutes cannot pass through the membrane, so instead the water is going to move through the membrane so that it equals out the concentrations. So we have less solute on the left, more solute on the right, but what that really means in terms of water is that we have more water on the left because there's less solute, and less water on the right because there's more solute. So the water will move so that there's an equal amount, like the ratio between water and solute is equal now as the water has moved into the right. So different things can affect the rate of diffusion. Temperature, as things heat up, they move faster. As they cool down, they move slower. The concentration gradient itself, we talked about that a little bit earlier. If the concentration gradient is like really out of whack like this, you see you have so many on the left side, it's going to move faster than this will because it's almost balanced already. The surface area, this allows for like a lot of just like movement in because it's all very open, but this you have to kind of find the areas. If you had a lot of surface area though, obviously things can move through the cell membrane. If the more membrane you have, the more proteins that are available, the more that molecules can move through it. But if they fold in on each other, if this were to like fold in, you've lost that surface area. So it's a bit of like, okay, we like having more surface area, but it does need to be in the capacity that I can actually access the surface area. And then length of diffusion path, again, you see that this is like pretty straightforward. It's a really thin membrane. They can easily pass through, whereas this is really thick and awkwardly shaped. It's not all even here, so it's a little bit harder for the solutes to pass through. Now, active transport is just the movement of molecules against the concentration gradient, but with energy. Because we're moving against the concentration gradient, that means we are forcing the solutes to be uneven. We want that. So we want an excess amount of solutes on one particular side. See how this is uneven already? The molecules, there's 5 over here and only 3 over here. What active transport is doing is pushing these last 3 molecules over into the left-hand side. It's purposefully going against the gradient. This is important to maintain homeostasis sometimes. So you do this all the time in your body, but this is what you use a lot of energy for. So naturally, molecules want to be in equilibrium. So this is forcing like an unnatural activity, which is why you need energy. Again, these molecules need to bind to specific proteins to activate a pump. When that pump is activated, it will release ATP, which causes a change in the pump, and it will allow the molecule to move from one side to the other, allowing the concentrations to continue to be uneven. So how this actually happens is once you're here, well, this is talking about ATP, but this is how, like, an ATP will bind to a protein and then it will activate the release of energy, and it does that by releasing one of the phosphates because the energy is stored in the bond between the second and third phosphate. And so hydrolysis, which is just the breaking of the bond and the production of water, will break this bond, and then the energy will be released, which allows the protein to change its shape and move the molecule across the membrane. To get ADP, which is what this has now become, back into ATP, you just have to eat some food and then go through cellular respiration. So talking about how these pumps actually work, we sort of just talked about it, but we'll talk about it with this picture now. So we have whatever molecule we are trying to push through, we'll target and bind to a pump. So this little like red dot will go into this little pump here, and then ATP will also bind to this pump. Once ATP goes through that hydrolysis and breaks off the third phosphate, which is what I talked about on the previous slide, the actual pump will change shape and it will kind of like flip, right? So now it will close this side and open the other side and allow that molecule to unbind and be released into the other side of the membrane. Once this is all done, the pump will go back to its original form, which is being open on the original side. You have this in your body. An example of this is a sodium-potassium pump. We're going to talk a lot about the sodium-potassium pump, but that's just a really good example to kind of like get you started here. There are different types of active transport methods. I'm sure just looking at this picture, you can kind of figure out what these different types of proteins do. So we have a uniport, and if you know anything about the prefix uni, that means one. So uniport allows for one direction and one molecule to come through. And then cotransport is with multiple molecules that can go through. You either have symport, which means that they're both moving from one side to the other, or antiport, where you have one moving in and another moving out. Okay, so these prefixes, I'm sure you've heard them in other terms before, like unicycle, one wheel, stuff like that. These prefixes don't change their meanings just because you're using them in a science aspect. So if you recognize prefixes, they're probably going to be the same meaning. And then finishing up, we're going to talk about endocytosis versus exocytosis. So endo means inside, exo means outside. So endocytosis is a molecule moving into the cell. What it does is it pushes against the cell membrane, and it pushes enough that the cell membrane, because it's fluid, will kind of go around it to prevent itself from tearing. It will go around this molecule, whatever it is, and eventually will reconnect on the other side and create a vesicle around itself, and then that molecule will be able to travel into the cell. The opposite can happen. That's called exocytosis, and that's a molecule moving out of the cell. So it pushes against the membrane from the inside, and it pushes until a vesicle creates around itself, and then it can travel outside the cell. And then these two different cytoses are just whether you can ingest solid or liquids. So phagocytosis is the ingestion of solid molecules, and pinocytosis is liquids and solutes. So if I had, for example, a large sugar coming in, that is a solute, so that would be pinocytosis. Okay. And then the last thing is talking about specifically protein exocytosis. So proteins are produced by the ribosomes of the rough endoplasmic reticulum, and they enter the lumen, which is just the inner space of the endoplasmic reticulum. When it exits the endoplasmic reticulum, it enters the cis side of the Golgi, and at this point a vesicle is involved. And as the protein moves through the Golgi and exits on the trans side of the Golgi, it is inside a vesicle. Now this vesicle with the modified protein moves and fuses with the plasma membrane, and then it will push against the plasma membrane until that vesicle is allowed to leave the cell, and it will be able to travel to wherever it is that it is planning on going, another cell, a muscle, whatever it may be. So the idea of protein exocytosis is the secretion of a protein from a cell to somewhere else in the body. All right, great job. We'll practice this more in class. I'll see you then.